Let's look at this diagram. Showing here is an alpha helix and its imaginary axis. And the red dotted lines here are the hydrogen bonds. So it is formed between an amide group and a carbonyl oxygen in a N+4 fashion, meaning that the first amino acid residue is linked to the fourth amino acid residue. So if we count from this amide nitrogen, which is N+1 would be this one, N+2 would be this nitrogen, N+3 followed by N+4 would be the carbon atom. From the carbonyl group, they will form a hydrogen bond with the N residue. So in average, there are 3.6 amino acid residues in a single turn of the alpha helix, which also means that the smallest structure of alpha helix possible can be just composed of 4 amino acids. So you can imagine that hydrogen bonds sort of like acting as springs in this structure, maintaining the position of alpha helix in place, and they are parallel to the axis of the alpha helix. And now, where are the amino acid side chains? Well, all these side chains are facing outward with a separation of 100 degrees in between these side chains. Why is it so? Because there is 360 degrees and you have 3.6 residues in one turn. So simply divide 360 by 3.6, you will get the answer of 100 degrees. Okay, you might wonder if the helical structure of protein is similar to the DNA helical structure. The answer is no, because in DNA you have a double helical structure, which is composed of two strands of nucleic acids. While in the protein alpha helical structure, you only have one polypeptide chain, so it is much smaller than the DNA double helix. However, some alpha helices do bind to DNA double helix. So these proteins are known as DNA binding proteins. The binding of such proteins control gene expression, and they are very important targets in cancer studies. So guess what? Both type of helical structures are interacting to one another by hydrogen bonds, which are in between the side chains of the proteins and the nucleotide bases of the DNA.